Hello everyone, how are you doing? I hope everyone is doing good. So welcome to another video of molecular basis of inheritance. This is chapter 4 for CBSE and chapter 5 for chapter 4 for Maharashtra board and chapter 5 for CBSE. One of the very important chapter. In this chapter we are discussing about the molecular level. Okay, we are at the molecular level of inheritance. That is, we know that DNA is the genetic material now, because in the previous video we have covered different experiments carried out by Griffith, Avery, McLeod and McCarthy, Hershey and Chase, who went on to prove that DNA is the actual genetic material. And in lower standard, you have studied about the structure of the DNA in detail. Also, we have covered the DNA packaging in detail in the previous video. If you have still not checked out the video, please check those videos out, uh, because now we move on to another important topic of this chapter, that is DNA replication. Okay, and from the DNA replication it was found out that Michelson and Stahl experiment, to these were the scientists who found out the semi-conservative nature of replication. What is the semi-conservative nature of replication, that we will see after understanding DNA replication. Okay, so let's begin. DNA replication. Replication means what? Creating a copy of its own, right? You have studied cell division like mitotic cell division, meiotic cell division. What happens in that? The parent cell divides and gives rise to two daughter cells, right? With equal amount of genetic material. So this genetic material which the parent cell has, it first creates a copy of its own. So you have diploid number of chromosomes. So first it undergoes making a copy of its own. So the amount of genetic material, the number of copies of the chromosome increases by twofold, 4n, and then cell division will happen. So equal amount of chromosome will go into both the daughter cell. So what are we going to do in DNA replication? How this 2n converts into 4n? What is the uh, mechanism involved behind this replication? How the Xerox happens inside the nucleus with regards to all the enzymes and the steps that are required in making a copy of the parent DNA, that we are going to see. You have studied the cell division, this part of the story, in lower standard, how the parent DNA having 4n material divides into two daughter cell in the mitotic cell division, or if it's a gamete cell, then meiotic cell division. Now we will see how this 2n gets converted into 4n. Okay, how the parent DNA makes another copy of its own. Okay, and then equal amount of DNA can be distributed into daughter cell. So this process is known as DNA replication, making a replica of the DNA. Okay, so we will consider a small segment of the DNA. In DNA packaging, we know that the simplest structure of the chromosome is the DNA. Yes, okay. So the simplest structure of the when you open up the whole chromosome, we studied the eukaryotic packaging and the prokaryotic packaging. Mostly eukaryotic packaging we will be keeping in focus whenever we are discussing DNA replication and further topics like transcription and translation. So this DNA replication, yes. So the chromosome is a result of multiple packaging that took place. Okay, so the two double helical structure first is wrapped around the nucleosome, then the nucleosome wraps around its own self, the chromatin structure, read on string structure, then the chromatin structure, solenoid structure, chromatin structure, chromatid, and finally the chromosome. So we will go to the simplest form, simplest molecular structure, that is when you open up the whole chromosome, the last level, okay, the initial level of the chromosome is your DNA. After this is the last, arrangement, molecular arrangement of the genetic material. So you have two strands. Yes, here for our ease of understanding, we will always represent strand as these two parallel lines, either like this, standing or horizontal. Otherwise, initially it is double helical structure, okay, like this, twisted and torn. Fine. So for our understanding, we will always represent two parallel horizontal lines or two parallel vertical lines. Fine. So what are the steps involved in it? The first step is initiation. Initiation means start of the replication process. So to begin any replication process, there will be a start site. Simple to understand. There will be a start place or an origin place. For example, a race has to be run. So there will be a start spot and end. So here all the participants are standing, and then from here they will start running to the end, to the finish line. So this starting point is known as the origin of replication. Ori, you can also refer it to as the Ori. Okay, so DNA replication starts at a point called Ori site or origin of replication site. Okay, a unit of replication with one origin is called a replicon. So one origin site, this particular unit is known as replicon. Okay, so initiation is brought about by now, so the start site. Then we know that the DNA is twisted and coiled. Okay, it is twisted like this. So this twist has to, this double helical structure has to open up, and it has to release the twist which is there in this helical structure. So this enzyme known as helicase. So the first enzyme which will come into participation, what it will do, it will open up. As you can see, let's say this picture over here. This helicase will unwind the two strands and separate the DNA by breaking the hydrogen bond. So the coil has to open also, and both the strands which are held together by hydrogen bonds, they have to open up. They have to separate from one another so that the copy can be made. Okay, so this is your DNA, parent DNA. It has to open up like this. Okay, so that the enzymes can come in and make the daughter strand. Fine. So you have two enzymes that will participate. The first one which will open up this zip is known as helicase, and the another enzyme which is known as topoisomerase, this will open the coiling. This will open the twisting and coiling. Okay, so if I draw a diagram like this, for example, right? So helicase will come over here in between the strand, and it will open up like this. It will broaden up the two strands like this. Helicase will open in between. It will act as unzipping the double helical strand, and the twist and coil which are there ahead. So that twisting and coiling will be straightened up by the enzyme topoisomerase. So you see ahead of helicase is topoisomerase. So this will keep the strand ready without any coils or without any twist. Like you can imagine the telephone wire. You have the telephone over here, and then you have the old landline setup. So the wire is, you know, coiled like this. So topoisomerase will be over here. Topoisomerase will open up this coiling ahead. Okay, and the helicase enzyme will come after that. Okay, so helicase enzyme will open up. This is the helicase enzyme. You can see the direction. It will open up the two strands by breaking the hydrogen bonds between them. What will be the hydrogen bonds existing? AT and GC. Okay, so these bonds will be broken up by helicase. The name itself is telling you helicase, breaking hydrogen bond and separating the two DNA strands. Okay, then these two separate strands will act as now as a template. Just like you go for Xerox for a Xerox person, and then you have a Xerox machine, right? And you give your document, and that document act as a template, and another copy of that document is made in that Xerox machine. So in that similar manner, the original document will be your template that will serve as the template for the synthesis of new strand. So this is your parent or the old strand. Now new daughter strands will be made corresponding to the sequence which is there on the parental or the template strand. We call it as a template strand. Okay. Now this DNA helix has a tendency to come together again. This helicase, as it is moving forward, this these two strands which were separated by helicase, they have a tendency to come together again. So as and when helicase moves forward, so the behind portion might come together again. Okay, so till the replication happens, you have single stranded binding proteins. You can see over here in this picture, this gray colored protein molecules. These single stranded binding proteins will keep what? They will hold these strands separately till the replication, till the daughter strands are being made over here. Till the daughter strands are being made over here. It will prevent them from joining together. Once the replication, once the daughter strand on both the template strand has been made, so they can come together again. But by the time new strand, new DNA molecule is getting prepared over here, to keep them separated, single stranded binding proteins will be required. Okay, so single strand binding protein, some individual proteins on one strand, another set of proteins on the another, they will keep the strand separated for a certain period of time. Helicase will break the hydrogen bonds and topoisomerase will release the twist and coiling ahead of the helicase. Okay. And yeah, topoisomerase prevents supercoiling and partial opening of the helicase. Helix forms replication fork. So for a short period of time, this double helical structure which was there twisted and coiled will open up for a certain period of time till the replication happens. This is known as Y-shaped structure if we represent like this. So this Y-shaped structure is known as replication fork. Partial opening of the helix forms what? Something known as this Y-shaped structure like a fork is known as replication fork. So this happens in the initial stages of the replication process. The recognition of the start site, there the helicase enzyme will come, open up the strands. Topoisomerase is keeping the strand ready without any coils or twist. Single stranded binding proteins will come and it will keep the strand in place till the replication happens. New strands are formed. Okay, like this you can see. Fine. Now, one more thing you can see over here in this picture is that the RNA primer and the enzyme primase. Okay. Now, to start the formation of the new daughter strand, to start the formation of the new daughter strand, you require a start point. So, let's say the origin starts from here. So, first an RNA primer will come over here. Now, you will say we are making a copy of DNA from where RNA primer comes. So, this is how nature is there. For the DNA to start, there will be a short stretch of RNA primer first. Like you have, imagine a train is there. So, sorry. So first the engine is there and then you have all the coaches, right? Then you have all the coaches, right? So this engine to begin the train is the first, first box. So this engine is nothing but the RNA primer. It will start the addition of the nucleotides over here. We know that the DNA is made up of nucleotides. So how this replication will happen? This engine will bring one coaches over here. One coaches means what? One nucleotides. One nucleotide, another nucleotide, another nucleotide. So these nucleotides will be brought and put forth over here. And then it's like all the coaches, like one full tray will be formed. Okay. Afterwards, at the end, this RNA primer is of no use. Once the whole strand is made, this RNA primer will be removed. That we will see at the incoming slides. Okay. This RNA primer will be first attached to begin the addition of nucleotides over here with the help of enzyme primase. Primase will bring RNA primer and join it to the beginning, to the origin site from where the replication process will start. Okay, so this, it will look like this. So DNA unzips, part by part as and when the replication fork is moving forward. Okay, now you need to remember four words over here. As you can see over here, leading strand, continuous, lagging strand, discontinuous. Okay, the replication will always happen in 5 prime to 3 prime end, from 5 dash to 3 dash. As you can see over here, from 5 direction, from 5th 5 prime to the 3 prime end. Just remember this, memorize this forever. Always the replication will start in 5 prime to 3 prime end. What is this 5 prime and 3 prime end? This we have discussed in the previous video. So if you have not checked out that video where we were covering the recap of the structure of DNA, there we have discussed this in detail. What is this 5 and 3? Okay. Also, you had covered this in lower standards, 10th and 11th standard. So the direction of synthesis of new daughter strand will always take place in 5 to 3 dash. So we know that the strands of the DNA are having anti-polarity, anti-parallel in nature. Okay, so if one strand is into 5 dash to 3 dash direction, another strand is in the direction of 3 dash to 5 dash. That is how both the strands will be made, daughter strands, but they will be made in opposite direction. They will be made in opposite direction. So this strand goes the formation process like this. Okay. So one strand you see moving in this direction, another in this direction. Also, one thing you notice over here is this strand is made in one go without any breaks. Here you see breaks are there, small fragment, there's small fragment and small fragment. That is why it is known as discontinuous. This is naturally, it was discovered by a scientist known as Okazaki, and he found out that both the strands of the daughter new strand that will be made out of the template strand, one strand is made continuously without any break, another strand is made in parts. So that is known as the lagging strand, and the one which is made in one go leading strand. There will be a short difference of time lapse between the two. This will be made at a faster rate, and the other will be made at a slower rate. Okay. Okay, so this will be known as a leading strand, which is made continuously, and this will be known as lagging strand, which will be made in short fragments. And then afterwards, the short fragments will be joined by an enzyme. Fine. So moving forward, a recap of whatever we have discussed up till now. The replication will always begin from 5 dash to 3 dash direction. Okay. So because of the anti-parallel nature of the DNA, the daughter DNA will also be formed in anti-parallel form. Fine. So to begin with the replication process, primase enzyme will first come. That is nothing but RNA polymerase. It will bind a small RNA primer to start the replication process, and then nucleotides will be added. Now, this nucleotides will be added with the help of which enzyme? DNA polymerase. Very, very important. You need to remember this. As I told you, RNA later on will be removed and replaced with DNA. Okay. So you have the RNA primer, and then now one nucleotides will be added. So to add the nucleotide is what? It makes up the DNA. So which enzyme will be required? DNA polymerase to make a polymer of the DNA. Okay. So DNA polymerase will do what? It will add new nucleotides from the 3' end of the primer complementary to the template and form a nucleotide chain. So if this is the template strand, for example, okay, this is a template strand, as you can see, let's say this strand, and the sequence is A, T, T, let's say G, G, okay, and the daughter strand which will be made now, what will be the sequence? Which nucleotides will be brought? A pairs with T, so thymine nucleotide, then A, T pairs with A, then A, G pairs with C, and G pairs with C. So this will be the sequence of the daughter strand. Okay, these nucleotides will be brought in one by which enzyme? DNA polymerase. Okay, fine. Then leading strand, as I told you, only one primer will be attached and a whole stretch of DNA will be made. This DNA will be made by which enzyme? DNA polymerase. You can remember DNA polymerase 3 as the name. Okay. Then it is synthesized in a continuous manner because it is known as the leading strand from 5' to 3' direction. Then lagging strand, it will be made in short fragments. Those fragments are known as Okazaki fragments. Okay, so short prime RNA primers will be there, and they will make short coaches of the train. You can just imagine like that. And here the synthesis of DNA is discontinuous. Okay, so then at the end, once the synthesis of the new DNA strand has taken place, as you can see over here, then these two strands will be the new daughter's DNA, and then the parent DNA will regain its original structure. Once the lagging and the leading strands are done with the formation of the new strand, as you can see over here, leading strand, one shot of primer, one stretch of primer, and this whole green thing is the new daughter strand. And your Okazaki fragments, that is discontinuous strand or the lagging strand, made in short fragments. This RNA primer will be later on removed by the enzyme DNA polymerase 1. Okay, it will be removed and replaced by DNA only. So these short stretches will be filled in by the nucleotides again by the same by the enzyme DNA polymerase 1 and Okazaki fragments. So once the DNA polymerase adds the nucleotides, there will be certain gap between the backbone, sugar phosphate backbone. So that will be joined by the enzyme DNA ligase. So DNA ligase will seal the gap. For example, this is one Okazaki fragment. This is one Okazaki fragment. So RNA primer has been removed, and these gap will be sealed in by which enzyme? DNA ligase enzyme. Okay, so you can have a look at the diagram over here. So this is the whole process of DNA replication. So you have the helicase coming in, then single-stranded binding proteins. Helicase will break the two strands, separate the two strands. Then DNA polymerase will come in. These are the enzymes. DNA polymerase will add nucleotides, and you have the RNA primer, and then DNA polymerase again bringing nucleotides and making the daughter strands. Helicase moves forward step by step. Then again RNA primer. So this is how in short fragments the DNA daughter strands will be made, and single-stranded binding proteins will be held. Will keep the two strands, template strands, away from each other till the replication is happening. Once the replication is done, they will coil back. They will form the double helical structure. This is how the whole process will take place. Then RNA primase will come. It will remove the RNA primer. DNA ligase will come and seal in the gap. This is the leading and the lagging strand. Okay, we'll do a recap once more.